In this chapter, we're going to explore things in populations or population genetics from that perspective, which is looking at genetics a little bit differently than what we've been doing so far. We're going to talk about how the population gene pools are defined, and also define a polymorphism, and a little bit of an introduction to some of the kind of math that we might be doing in terms of alleles and genotype frequencies. So first, let's think about what genes are. And really, that's a very simplistic question, and I would answer that with it depends on your view. So if you are a transmission geneticist, and really you think of a gene simply as a unit of inheritance. If you're into molecular genetics, you really think of a gene as a macromolecule, this polymer of nucleic acids. Whereas if you're a population geneticist, you're thinking about genes in a population and how those genes behave over time in terms of the numbers or the frequencies of them, and what happens to them as time passes. Now, another thing I want to remind y'all of is, what is a population? So a classic definition of that is a group of interbreeding individuals in a given location and at a given time. Now, populations can change, and they do. And these are three major ways they can change. Their size can fluctuate, can go up, can go down. Their geographic location can shift. And I'm not talking about necessarily large movements, but even small movements can have huge impacts on populations. And then ultimately, the genetic composition can change. And we'll talk about some other ways that you can manipulate the genetic composition via selection and migration. But also, these kind of things, size and location have a big impact, or can potentially have a big impact on the genetic composition of a population. So when we're talking about genes and populations, conceptually, what we're thinking of or what we're focusing on are all the alleles of every gene in that population, and those make up the gene pool. So sometimes you'll hear people say, "you out of the gene pool." That means they're not going to be reproducing anymore. Not just that you might want them gone. So in this case, reproduction is critical. So those individuals that reproduce are going to contribute to the gene pool of the next generation. So we're always kind of thinking about the next generation after. So we can think about alleles of a gene. Remember, you can have many alleles. In any one individual, only 2 alleles are possible. That's the maximum number of alleles because we're diploid individuals, and I should qualify that with a diploid organism. So, however, we know that there can be many alleles in a population. Just think back to our ABO blood group work, and we saw multiple alleles with that kind of a situation. Even with beta globin, they've been worldwide, they've identified over 600 different alleles for the basic beta globin gene. Now, population geneticists look at this genetic variation that's present in the gene pool and then examines how it's going to change from one generation to the next. So the previous discussion leads us to the defining what genetic variation is. In general, population geneticists focus more on mutations as a new source of genetic variation in populations. And we can divide those into the 3 main types that we've talked about before. We can have chromosome mutations. Those are where you get those structural variations, i.e. the inversions, the translocations. Those are different types of mutations, but at a larger level than what we're commonly think of in terms of mutations. Genome or genome mutations, those are in dealing with the numbers of sets of chromosomes or numbers of chromosomes, either cases of euploidy or aneuploidy. In terms of the gene mutations, what we're referring to here would be like point mutations or deletions or additions of nucleotides at that gene level. Now, there are other sources of genetic variation. There can be recombination. So you can get new combinations of alleles in your gametes. Sexual reproduction is a big source of genetic variation via fertilization. In this case, you can get new combination of chromosomes via the fusion of these gametes. You can also introduce new alleles to a population. That would specifically be via migration. And then there's a bunch of other ones that we don't necessarily go into or we won't talk about, but we have talked about one in the sense of just transposable elements. Transposable elements can shift exons around. This doesn't tend to be an immediate kind of generation of genetic variation. It would be over long-term, like evolutionary time. There's also horizontal gene transfer. That is seeming to play a very large role in the amount of genetic variation that we see in populations. Again, this is more of an evolutionary time frame. But I want to point out that here, maintaining that concept of the gene pool and what population geneticists focus on, remember that all individuals in a population may not reproduce. So you can have mutations that would be lost simply because those individuals did not pass any unique alleles on to the next generation. Now, a few more terms to get through. In terms of genes, we would use these kind of terms for the population level. For example, a gene could be polymorphic. In that case, we've got many morphs, if you will, poly being many. In that case, we've got 2 or more alleles in the population. A monomorphic gene means that you typically only see a single allele in a population, and this does happen in certain cases. Now, variation, that's more at the gene level. You can see variation within a given gene, and these are classified as single nucleotide polymorphisms. So there are single nucleotides that account, or differences in those single nucleotides that account for approximately 90% of the variation between individual people. They're just single changes, like a transition or a transversion. In humans, that would mean that a gene that is 2,000 to 3,000 base pair contain up to 10 different polymorphic sites on average. SNPs are used quite frequently in different types of genome-wide association studies to identify the causes of diseases, and also very frequently in population studies. Now let's look at some math. It's kind of basic, just some basic ideas that I want to get across to you guys. And there's 2 ways you can estimate allele frequencies. One is from direct counts. So in this case, we've got a 2 allele system, i.e. we've got a green allele and a purple allele. So most of what population genetics is going to focus on are these allele frequencies, and then also look at how alleles come together to form the genotypes, because there's some ways that you can change those associations. Now, simply with allele frequencies, if you're given a whole bunch of alleles, where you have n of 100 alleles, we can count up the frequencies of each of these respective alleles. For example, there's 60, there should be 60 of the green, there should be 40 of the purple, divide by 100, which is the total number of alleles in this population, and that gives you the frequency of the green allele versus the purple allele, where the purple allele will be 40% in this case. Very simplistic kind of idea. Now, if you have actual genotypes, if you go in and you count the genotypes, again with a 2 allele system, I just want to first point out here that on the last one we had 100 alleles. Technically, we only had 50 individuals, right? Because every individual would have 2 alleles in a diploid organism. In this case, we have an n of 100. This is referring to the numbers of individuals that are present in the population. So how many alleles would we have in this? Simply 100 times 2, that would give us 200 total alleles in this population sample. Now, there's another way that we can estimate that too, or look at that. If we count up all the numbers of individuals that are homozygous for the green, we should get 36. So 36 divided by 100, which is the total number of individuals, gives us a genotype frequency of 36%, or 0.36. We can also look at these 2 classes of heterozygotes. And we're separating them out because there's 2 different ways that you can produce a heterozygote. You could either get the green allele first and then the purple allele, or the purple allele and then the green allele. And these can be different in terms of their identity by descent. So the frequency of the heterozygotes, the total frequency of heterozygotes in this population is actually going to be 48. So 48 divided by 100 would be 48%. For the individual genotypes of purple and green, we've got two different ways that we can produce those. And then you can also count up all of your purple genotypes. That would be a total, there should be a total of 16 in here, and 16 divided by 100 gives you 0.16 or 16% in terms of the frequency for that genotype. So these are 4 different genotypes, 2 of which end up potentially being the same, but they're slightly different in terms of which allele they've received and in what order. Now, we can take these numbers and actually estimate the total number of alleles in the population also. And let's look at just the frequency of green in this population. We notice that here we've got 2 alleles. Well, we've got a total number of 36 individuals. We multiply it by 2, and that gives us the total number of green alleles in those individuals that are homozygous for the green allele. Then we've got 24 individuals that have a green allele from this class of heterozygote. And then we have another 24 that have the green allele in this class of heterozygote. So that should give us a total of 120 green alleles in this population. Now we can also do that for the purple allele. We can go through and calculate frequency of purple. Again, that's going to be 16 times 2. That gives us the frequency of the purple allele in these homozygotes. Then we've also got to account for the purple alleles that exist in these heterozygotes. So that'd be 24 plus 24. That should give us a total, if I did my math right, a total of 80. We can sum up 120 and 180. And as a check, that should give us 200 total alleles. So these are 2 different ways that you can look at calculating the frequencies of alleles in populations.